This is Module 12, which is our final module in Chemistry 110. In this module, we'll talk about nucleic acids and their role in transmitting the genetic code. When we talk about nucleic acids, we're primarily concentrating on DNA, which is deoxyribonucleic acid. It consists of three essential parts: a sugar, specifically deoxyribose, one of the nitrogen bases, either thymine, cytosine, adenine, or guanine. It's a double-stranded molecule, and we'll talk about the structure in a second. The other main nucleic acid is RNA, which is ribonucleic acid. As compared to DNA, RNA contains ribose instead of deoxyribose. In the four different nitrogen bases contained in RNA, the thymine of DNA is always replaced by uracil. Adenine, guanine, and cytosine remain, and unlike DNA, RNA is single-stranded. Here's the structure of the backbone and the constituents of a nucleotide. Each of these essential units contains a sugar, either the deoxyribose in DNA or the ribose in RNA. The pentose sugar is connected to a phosphate group, and it is also connected to one of the nitrogen bases. The pyrimidine bases are cytosine, thymine in DNA, and uracil is its thymine's replacement in RNA. The purine bases are adenine and guanine. And the assembly of the polynucleotides connected by the phosphate to sugar to phosphate groups are shown. The primary structure, the sequence of nucleotides in DNA or RNA, is usually read from the 5' position of the sugar. That's the place where the phosphate group is connected, and the nitrogen base connected to the other part of the ribose or deoxyribose, and then to the following 3' positions. So the sequence of nucleic acids, like the primary structure of amino acids, is listed from the 5' phosphate to the 3'. So in the illustration shown here, we're going in a downward slanted direction, going from 5' to 3'. So we would read this as guanine, cytosine, uracil, and then adenine. GCUA. Some of the early studies on DNA showed that for every adenine molecule in DNA, Chartrand's rule showed that each adenine was matched by an equal number of thymines. In the same way, each guanine was matched by an equal number of cytosines. So these two pairs always link to each other. The structure of DNA was finally found in the early 1950s by a team consisting of James Watson, Francis Crick, and Rosalind Franklin. By crystallizing the DNA molecule and shining X-rays on it, they were able to calculate the structure as that of a double helix. Those two backbones intertwined amongst each other, where one strand is in the 5' to 3' direction, and the complementary strand is in the 3' to 5' position, with each adenine connected to a thymine in DNA and each guanine connected to a cytosine. These are known as base pairs, and they're held together by sequences of hydrogen bonds. In adenine and thymine, there are 2 hydrogen bonds holding that particular base pair together. Notice that in guanine and cytosine, there are 3, so the assembly of GC is a little bit stronger than the others. DNA replicates by causing the 2 strands to come apart, and then the complementary nature of those bases attracts new nucleotides to the assembly, complementary nucleotides, and watch the YouTube video to see how this operation occurs. DNA replication starts with an enzyme called helicase, which unwinds the strand. DNA polymerase transcribes, that is, attracts new nucleotides that are complementary to the leading strand. The lagging strand, known as Okazaki fragments, are filled in with other complementary nucleotides, thus forming from the original strand 2 of what are known as daughter strands. In order to translate the information contained into DNA, a number of different types of RNA molecules have to attach to the DNA template strand, and they transcribe that section. Here's how DNA transcription onto RNA occurs. The segment of double-stranded DNA lines up with the RNA, but notice that not every particular nucleotide is translated. There are segments of DNA that do not code for important proteins. These are known as introns. And notice that when the DNA starts to replicate, they form little loops so that they're not contained into the information. Segments known as exons code for protein synthesis. So when RNA connects to DNA, we have the two strands, the DNA information strand and the DNA template strand. What is the DNA sequence corresponding to the DNA information listed? Well, remember that the two strands of DNA, the informational strand and the template strand, run counter to each other. Each A links up with a T, each G links up with a C. So here are the two complementary strands. When we're trying to find the necessary RNA transcription strand, the messenger RNA, notice that it is complementary to the template strand, but as compared to the informational strand, they're almost exactly the same, except that the DNA informational strand as compared to the RNA, messenger RNA strand, each of the thymines have been replaced by uracil. So directly from the DNA information strand, we can figure out what the necessary messenger RNA strand is. The messenger RNA takes the message from the DNA, but in order to build the necessary protein coded for by that DNA, we're going to need transfer RNA, tRNA. Here's the shape of a transfer RNA molecule. The codon, notice that the very base contains a series of 3 bases. In this particular case, they happen to be GCC. The complementary anticodon on the T transfer RNA is the complementary set of bases, and in doing so, the transfer RNA can then, by the anticodon represented, go and find the appropriate amino acid coded for by that particular segment of messenger RNA. A codon is a 3-letter RNA triplet, 3 bases, nucleotide bases on the messenger RNA, and it turns out that there are a total of 64 possible codons which code for 20 different amino acids. Now notice on this list that a number of codons code for the same particular amino acid. This is a certain amount of redundancy, but it shows that even if a mistake is made, even if one of the nucleotides is incorrect, for example, in the case of leucine, we have 6 tries at it. There are 6 different codons for leucine. So now we can introduce the genetic code. We can figure out what tripeptide is coded for by the following messenger RNA sequence. The sequence is 5' GGC, CUC, ACC. We can use that code on the previous slide to interpret what that tripeptide would be. And notice that the tripeptide is formed in the order from the nitrogen terminus to the carbon terminus. GGC codes for glycine, CUC codes for leucine, ACC codes for threonine, and then we have the carboxylic acid. So we've broken the genetic code. We know what tripeptide is formed by that particular sequence in messenger RNA and the resulting informational strand of DNA. Mistakes occur, and the redundancy within the genetic code sometimes doesn't eliminate the possibility of mutations occurring. Mutations occur due to changes in the nucleotide sequence. This can occur because of possible substitutions, or if a particular nucleotide has been deleted or inserted. We can use the 3-letter genetic code by analogy to this sentence, "Our big red dog bit the old man." A mutation can occur in a variety of ways. If we add in an extra symbol, notice how it garbles the message. No longer does the sentence make any sense. Likewise, if we delete one of the bases, the same type of garbling occurs. This is the type of mutation that occurs in the case of sickle cell anemia, which we talked about in Module 10 with amino acids and proteins. In this case, we said that the sickling part of the red blood cells occurs because of a change from a polar amino acid, glutamic acid, and this is an acidic charged amino acid side group. A mutation occurs by switching the DNA sequence from a T, a thymine, to an adenine. The result is, instead of a glutamic acid in the primary sequence of the protein, a valine, a nonpolar amino acid, is inserted. Notice the change in the shape of the red blood cells, and we previously talked about the increased propensity for excessive clotting, which can lead to situations like strokes. Surely you've heard about recombinant DNA, which is the insertion of a gene, a sequence of DNA from another species into another species. Usually, this is done by the use of plasmids, which are small circular DNA segments in bacteria. They're inserted, excised section of DNA is inserted into the genetic sequence, into the plasmid, which is then reincorporated into the bacterial nucleus. And as the bacteria reproduces, the proteins coded for by those cells are produced. This is one way that we're producing insulin in the modern day. The gene for producing human insulin is inserted into a plasmid. The plasmid becomes a recombinant bacterium. As the bacteria replicates, human insulin is produced. Prior to this, many years ago, diabetes sufferers had to take in pig insulin, which, as we've seen before, is the same as human insulin with the exception of one particular amino acid, which can cause some disruptive issues. This is a much more safe way of producing insulin for diabetic sufferers. Viruses. Surely you've come into contact with a virus or two in your lifetime. Influenza, cold viruses, hopefully nothing more than that. Viruses are materials that have nucleic acid, either DNA or RNA, covered by a protein shell, and the structure of a typical type of virus is shown here. What they are is little machines for reproducing themselves, and they do so by attaching to certain host cells. Viruses can only attack certain kinds of host cells. What they do then is insert their own genetic material and cause the host cell to reproduce. As the host cell reproduces, it fills with viral particles, the host cell bursts and spreads the infection. A specific type of viral cell that contains RNA is known as a retrovirus, and probably our best exposure to that is the human immunodeficiency virus, HIV, which is responsible for the condition known as AIDS. In this case, the RNA inside HIV infects host cells. In particular, these are immune cells, T cells, and causes them to become debilitated and make new HIV cells. This is an example of what that cell looks like, and there are different ways we're now approaching to limit the reproductive capacity of these retroviruses. This is our last module. In this module, we talked about DNA structure, replication, translation, and transfer. We've talked about how the genetic code codes for protein synthesis, when mutations occur in the genetic sequence, and we talked a little bit about viruses and retroviruses. We're at the point now where you should be reviewing the practice final for preparation for your final exam.